Hello and welcome to this video on the genetic differences between an ale yeast and a lager yeast, or the most basic degree of separation in beer brewing. The functional differences between an ale and a lager are fairly well known. Ale ferments warm and at the top of the wort. Lager ferments cold and at the bottom of the wort. Things are more complicated, but this covers the generally recognized distinguishing features. The question is what makes these and other differences. As with many things, we can trace this phenomena directly back to genetics. Admittedly, these aren't the fun kind of genetics, given that we go straight to Germany for the earliest data we know of. A lager beer emerged in 16th century Bavaria, and it was effectively a response to the regulations like the Reinheitsgebot that stated you could only do certain things when making beer. For example, beer could only be made from water, barley, and hops. So, naturally, certain yeast was used that was more amenable, let's say, to the use of these particular ingredients in the cold environment, especially since, to some extent, in the mid-16th century, beer brewing was legally restricted to only being done during winter. Because you had, obviously, cold environments and colder cellars, a top fermenting yeast, that is effectively ale yeast, really wasn't amenable to this scenario. The flocculation of yeast led to it appearing at the bottom of the fermenter, and yet you still needed it to function, rather than gathering all together. This is where the lager yeast strain comes in. The lager yeast is a little bit weird, mostly because it was really first used on a large scale in that 16th century, the 14th to 16th century, but you see it mostly emerging into the 16th century. It's a hybrid of two different yeast strains. Saccharomyces pastorianus, which is the actual lager strain that we know, that came about from the Saccharomyces cerevisiae, and a second kind called Eubanus. These two, Cerevisiae and Eubanus, led to Pastorianus, a lager yeast. The key thing here is that the Eubanus strain allowed for the Saccharomyces cerevisiae to acquire cold-tolerant genes or cryotolerant genes. This did lead up with, let's say, weird phenomena happening. For example, multiple copies of chromosomes ended up in the lager yeast versus the ale yeast, that parts of the genome were very much different, and others again were either lost or silenced. If you want to try and track down the origins of this, let's say, hybridization event, primarily the evidence indicates that it occurred in Germany and again in Bavaria, but the source material needed for it, that Saccharomyces eubanus, seems to have come from the New World, or for all intents and purposes North America and South America. There is evidence of it in the wild in Europe, but the key thing here is that that evidence really only shows that it's present in things like oak and spruce trees, and nothing to do with fungi, like yeast. So it's unlikely, although theoretically possible. The second theoretical pathway from which the let's say hybridization event could have occurred is via the Silk Road, with a fair amount of evidence, although again nothing definitive, showing a pathway from Tibet through to Europe, and the genetic similarities in this, the Saccharomyces eubanus, appear to be the highest degree of similarity found so far. Again, nothing definitive, but still at least something more reliable than what was known beforehand. There is a third possibility, and that is simply that either an extinct or undiscovered species of Saccharomyces eubanus may have been somewhere in Europe, and that's how the hybridization event occurred. Although, given everything said and done, it seems more likely the Silk Road is responsible for not just the extension and, let's say, enjoyment of plague, but also, in this case, lager. You can do the same thing in a laboratory setting too. You can hybridize Cerevisiae and Eubanus to create Pastorianus. So it's not like this is a hard thing to do, although it is somewhat more difficult than you would think. Understand, if you want to do it, let's say via sexual reproduction in the case of yeast, it's much more difficult. You want to do it using genetic modification, it's actually very easy. The reason why this event probably has occurred, though, is again going down to that whole, there were laws meaning you could only brew in winter. Winter was cold in Germany, and therefore only the cold tolerant species or anything that hybridized to get cold tolerance would have survived. The chromosomes of your lager species are actually kind of weird. First of all, there is a strain-dependent copy number of a whole bunch of different chromosomes. So different kinds of your lager yeast will have different chromosomes. And some of these are not going to be the same on each side of the chromosome. You're actually going to have very different versions. One reason for this is that not all lager yeast is actually lager yeast, or at least not the Pastorianus species. There was a, let's call it convention, that meant that a whole raft of wild yeast strains that were found were simply thrown under the classification of being Saccharomyces pastorianus because they were wild yeast. Obviously, this is not a particularly good approach because you end up with things belonging in a different group and coming under this umbrella. This may also explain why we have a very, let's say, wide raft of different genetic types of lager yeast. The, let's say, consistency or homogeneous nature of Cerevisiae strains is much better. In short, Pastorianus is very wide and diverse. Cerevisiae, so ale yeast, is generally going to be much more consistent and reliable. You can look at this by basically taking any kind of yeast you want, whether it is the commercial varieties, something from a craft brewery, something from an ancient brewery, or a wild type, and comparing the various products from it. This is not done so often, but it can be used for creating hybrids in the laboratory setting. In fact, one of the more interesting experiments, let's call it, shows that if you hybridize a yeast strain this way, they tend to have a better ability to adapt to their environment. Essentially, it's the same sort of thing with a human, and for that matter any other species, and that will evolve. If you put them in an environment where there is a selective pressure, and you allow them to reproduce, only those that are most fit for that environment will do so. This is a simple example of how we can easily get high ethanol tolerant strains of yeast, simply because you put them in an environment with a whole bunch of ethanol, and they survive. The other advantage of lager yeast, let's say, is that they tend to have a greater plasticity. That is, if you take a, let's say, culture, and you repeatedly use it, that is, you take the yeast out of your fermenter, you pitch it into a new fermenter, and you make more lager with it, there is a greater ability to avoid genetic problems. It tends to be more stable overall. Although it is worth noting that that's more stable, not that it is stable. It's an important distinction because you will still find that some of the yeast still gets messed up. If you were to use laboratory-made hybrids, it seems to be a little bit better again, but not necessarily so. The use of these hybrids, though, is not yet useful, but we expect to see it become increasingly useful, because you get things like amylases being produced in these hybrids, beta-glucanases, glucosides, and similar. The ability to use sugars that aren't obvious. So, for example, we've mentioned in other videos the use of lactose for milk-based alcohol. Most Cerevisiae species don't do this, but if you hybridize with another species, you can do exactly that. Or in other scenarios, you may want to change the amount or nature of what it is that's growing inside your particular thing. So if you want to get yeast that makes less diacetyls, well, one way to do this is simply to combine it with other yeast. On top of this, there are some big differences between ale and lager. It's notable that the two different, let's say, strains for now, have very different quantities and types of transporters for things like malt. Well, specifically maltose, which is the sugar within malt, but still, the differences between ale and lager yeast is very much apparent. There are 4 different types of transporters, let's call them. You have your MALX1, MTT1, MPHX, and AGT1. Now, it goes beyond just being a difference in the, let's say, presentation or production of these transporters on the ale and lager. Even within the surface of each of these, there's a notable difference. Generally speaking, Cerevisiae has a greater production of AGT1 genes, and they also have a greater number of MAL1 versions of the transporters. This means that they have a greater ability to take up things like glucose, which is unsurprising. Also for maltose. There's a certain amount of activity for maltotriose, but not as often. And finally, at least in terms of common sugars that are used, we have sucrose. The hybridized version of these, that is, for all intents and purposes, lager, have a greater number of the AGT1 and MAL genes. Theoretically, this should make them work better, because obviously more genes, more transporters. The problem here is that researchers found that about 60% of the genes that are there, at least in terms of the AGT1, have a mutation in them that means they have an early stop codon, producing defective genes and therefore the defective product. There are also other lager strains that appear to show this, but not so much ale. Because of this, and it's very important to be aware of it, glucose does affect this in terms of ale. Ale will be able to suppress or reduce the expression of AGT1 genes when in the presence of glucose. Next we have a comparison of the RTM1 gene. Typically speaking, you find this in particular strains of ale yeast. What RTM1 does that is particularly important and useful here is that it inhibits a certain, let's say, resistance or down-regulating processes in molasses. This allows for you to have either sucrose being used without any issue, or in other cases molasses being used without any issue, which is useful if you're, say, doing high-gravity brewing. Other research from about 30 years ago has found ways to make dextrin usable by yeast, which is useful mostly down to the fact that it means you can make low-calorie beer without having to really affect the taste as much. There is some debate around that, but the actual effect of it being low-calorie is not in contention. Then we have the wort itself, particularly when you're brewing beer as a whole. Lager seems to be better at using maltotriose than ale does, and it generally leads to less residual maltotriose at the end of fermentation. This is, generally speaking, down to certain transporters and genes again, so the use of greater amounts of maltose and maltotriose seems to be better. Ale is, let's say, genetically more agreeable to being dried, so those are pouches of yeast that you just pitch into your fermentation, and that sort of thing. And so it appears, at least based on the research, that certain genes make ale better for storage and easy distribution versus lager. The reasoning has been that the genes responsible for various things, but specifically intracellular trehalose, appear to be more active in ale than they are in lager. In terms of the, let's say, results, you're going to find that there is more sulfur dioxide in ale strains, as it appears lager has the right genes, and certainly more active versions of them, to avoid issues around sulfur not being metabolized, or at least not being metabolized fast enough. Combine this with the lower temperature of lager production, you do get less sulfur. We've already mentioned diacetyl, but there are other similar metabolites that are important, let's call them. For example, vicinal diketones. These are very similar to diacetyls, and they do have a similar, let's say, end result in that they don't smell particularly nice in excessive quantities. You also have alpha-acetolactate, and, well, that breaks down into diacetyl. There seems to be more concern around this in lager strains than in ale, and again, this is down to the genetics involved. The upside to lager is that a lager appears to be able to compensate for this later in the fermentation, where ale doesn't need to. And finally, we have the FSY1 gene. This is a fructose transporter. Lager strains do use this. Ale strains don't, which means theoretically, lager should be able to make use of, say, fruit juice or grape juice, anything similar, whereas ale can't as effectively. Theoretically, with the use of various enzymes to break down the fructose into, you might theoretically be able to ferment with it, but it's not entirely clear. Next we have your aromatics other than diacetyls, but including this. There is certain genes expressed, mostly related to the esters, although it will cover others shortly, and that this is down to the EEB1 gene in lager. There is a greater production of ethyl hexanate and ethyl octanate. Then we also have the ETR1 gene, although thankfully this doesn't seem to affect it, although it is present in lager versus ale to the same extent. And finally we have the ARO9 and ARO10 genes, which help to produce higher alcohol levels. Ultimately, it comes down though to the ILV5 and BDH2, which appear to be part of the same cluster of genes that show up in both strains when they do, that helps with the production of diacetyls. So it appears to be a trade-off in terms of lager. You get more alcohol, but you get diacetyl production. Further, you get certain other aromatics. For example, you have ferulic acid. This then gets converted to 4-vinyl glycol, and this is a phenolic compound that has a smoke-like flavor. Now, for some of these, you want this because it's nice and beer in that case, but you don't want it in others because it's very much not wanted. In cases where it is used, it's used primarily in lager because it adds a clove-like flavor, although not exactly, and that's why it's needed. This is why you need the FDC1 and PAD1 genes. They're present in the lager to a greater extent than they are in the ale genes. Then we have sake. Now, sake's sort of its own weird category, but it is technically seen as a beer, and you could certainly argue it as such, and you are going to have certain genes, such as TUM380, PAD1, and FDC1, that are very important in sake production. Admittedly, it also shows up in British beer styles, so take that as you will. We've mentioned already the importance of sulfur metabolism, and particularly the fact that lager produces more. Ale yeasts have a greater, let's say, appearance of the ATF1 and ATF2 genes. These promote ester synthesis and aren't responsible for alcohol, tobacco, or firearms. Esters are there basically to make fruity flavors and floral aromas. Your typical ale type sense and taste. Then there are flocculation genes. We mentioned early on that one of the things that made lager work the way it did was that during the hybridisation event, the flocculation was maintained, but other genes allowed them to be cold tolerant. This is part of why Cerevisiae has certain genes. Some of them are nearby to others are completely separate again. There are 5 genes for the most part, although technically 4 of them are sort of together. You have the FLO1, FLO5, and FLO9 all together, and FLO11 is way off elsewhere. These genes are there to allow for the production of adhesins and flocculants. These are what allow the yeast to bind together, hold itself into a nice neat mass, and sink to the bottom. Part of what makes this happen is the use of mannose residues or leftover sugars in the cell wall, and that allows for the adhesin to bind to it and act as a sort of intermediary between the yeast and the other yeast cell. What's important here is that the genes responsible for that are partly going to affect your things like mannose glucose use or similar, and the sensitivity to certain amounts of maltose, especially important in lager yeast, where, well, lager typically has a better utilization rate for maltose versus other similar sugars compared to ale. Ale will flocculate, mind you. It's not like this doesn't happen, but it's strange to say this, but ale has a lot more variability in the various flocculation gene expression, so it's not as easy to claim that it does one thing or the other, which is very similar to what lager is like with other genes again. It's also one reason why ale, typically in a traditional sense, ends up being somewhat cloudy. It's notable, as we mentioned, that some of the changes in ale and lager tend to be more useful than others, and one of these is in aquaporins. Aquaporins are transporters for, unsurprisingly, water amongst others. There is some weird shit going on when it comes to these, mostly looking at either frame shifting, that is, moving a gene, or deleting them, in other cases just mutating them altogether. And this has the effect of stopping them from working in a very general sense. The AQY1 gene tends to be broken, and this is found in wine strains rather than ale or lager as much, but it's basically not present in any beer strains, except for a couple where you find it, because they've been inadvertently put into the category of being a beer yeast. In broad summary terms of what we've described so far, lager yeast relies more heavily on things like the HSP12 and FAD3 gene for its cold tolerance, this being a result of it being hybridized with Saccharomyces eubanianus. This allows it to ferment at much colder temperatures, although admittedly not necessarily so. Further, the fermentation is not necessarily the most robust, but it does work well and reliably. By contrast, without these genes, you basically have a warmer temperature fermentation, and it does make issues when the temperature gets below 15 degrees Celsius or thereabouts. In terms of the, let's say, chromosomal part of things and genetic loss, there have been a very diverse range of these changes, but unfortunately they're so diverse and generally specific to a particular lager strain that it's hard to say this is exactly what you need to worry about. And by contrast, ale being generally and very broadly more consistent, you basically don't have to worry about this as much. There has been some mutation and gene transfer between ale yeast strains, but overall it's not as bad as lager. The primary genetic differences between lager and ale yeast mostly boiled down to the fact that in order to get lager yeast, you had Cerevisiae and Eubanianus hybridize, whereas with ale yeast it's generally just Cerevisiae. The advantages of the hybridization event are the cold tolerance and the use of things like maltotriose, and a stronger flocculation. Whereas ale yeast, in exchange for not necessarily being as cold tolerant or able to use as many sugars as widely and as efficiently, does ferment that much quicker, and to some extent produces much more varied flavors, which, given its genetic homogeneity, is actually rather funny. The genetic distinctions do lead to practical differences in brewing outcomes. For example, a lager tends to produce a cleaner, crisper beer, whereas ale tends to make a more, let's say, active and engaging beer style, meaning that when you drink it, there's a lot more differences. Although, then again, we would argue that the lager differences are just more subtle, but there's certainly a debate to be had there. Thank you for watching this video. If you found it interesting, consider liking, sharing, and subscribing, and please do post any comments, questions, or suggestions you have below.